Cell cycle is the name we give the process through which cells replicate and make two new cells. Cell cycle has different stages called G1, S, G2, and M. G1 is the stage where the cell is preparing to divide. To do this, it then moves into the S phase where the cell copies all the DNA. So S stands for DNA synthesis. After the DNA is copied and there's a complex extra set of all genetic material, the cell moves into the G2 stage, where it organizes and condenses the genetic material or starts to condense the genetic material and prepares to divide. The next stage is M. M stands for mitosis. This is where the cell actually partitions the two copies of genetic material into the two daughter cells. After M phase completes, cell division occurs and two cells are left, and the cell cycle can begin again. G1 phase is an intermediate phase occupying the time between the end of the cell division in mitosis and beginning of DNA replication during S phase. S phase is the phase of the cell cycle in which DNA is replicated, occurring between G1 phase and G2 phase. Since accurate replication of the genome is critical to successful division, the process that occurs during S phase are tightly regulated and widely conserved. G2 phase is a period of rapid cell growth and protein synthesis during which the cell prepares itself for mitosis. Curiously, G2 phase is not a necessary part of the cell cycle as some cell types, particularly young Xenopus embryos and some cancers, proceed directly from DNA replication to mitosis. So let's talk about the checkpoints. So there are nearly three checkpoints in a cell cycle. First check is metaphase checkpoint, check for chromosome spindle attachment. Next is G1 checkpoint, check for nutrients, growth factors, DNA damage. And G2 checkpoint for cell size and DNA replication. Thanks for watching. That's all for today.